Hello everyone, and welcome to today's The Scientist webinar. My name is Susan Harrison-Uy, Engagement Manager for The Scientist, and I will be moderating our discussion. Today, our panel of experts will discuss interactions between the brain and surrounding systems, including cerebrospinal fluid and the vascular system. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during the Q&A session. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. BioLegend develops and manufactures world-class cutting-edge antibodies and reagents for biomedical research at an outstanding value to customers. The broad product portfolio includes flow cytometry, cell biology, and functional reagents for research in immunology, cancer, and stem cells. In 2014, BioLegend added an experienced neuroscience research and development team, formerly Covance Antibody Products, with over 600 antibody products in areas such as neuroinflammation, APP and amyloid beta, tau, and synaptic function. BioLegend is certified for ISO 9001-2008 and ISO 13485-2003. Neuroscientist News is a centralized digital resource for research and clinical neuroscientists, providing timely coverage of news, publications, products, events, and conferences. We bring you exciting discoveries and insights into new trends and techniques in neuroscience and neurology. Our social pages on Facebook and Google+ foster an interactive community environment for learning and sharing. Connect with us today at neuroscientistnews.com. Our sponsors have provided us with some helpful resources, and we've posted these in our resource list located on the left side of the screen. And with that, let me introduce our first speaker, Dr. Maria Lentinen. Dr. Lentinen received her PhD in neurobiology from Harvard University. After a postdoctoral fellowship at the University of Helsinki, Finland, she returned to Harvard Medical School for further training in developmental neurobiology. Dr. Lentinen is currently an assistant professor at Boston Children's Hospital, Harvard Medical School, where the focus of her research program is to investigate the mechanisms by which the choroid plexus secreted signals in the cerebrospinal fluid regulate the development and health of the mammalian brain. Dr. Lentinen has received several awards and honors for her research, including an Alfred P. Sloan Fellowship and a Society for Neuroscience Janet Rosenberg-Trubach Career Development Award. Current research in her lab is generously supported by the Pediatric Hydrocephalus Foundation and the National Institutes of Health. Interested graduate students and postdoctoral fellows should contact Dr. Lentinen by email about research opportunities in the lab. Dr. Lentinen?
Okay, thank you, Susan, and thank you very much for the invitation to participate in this webinar. So as Susan very nicely introduced, the research in my lab is interested in investigating how the fundamental mechanisms that regulate the development and maintenance of the mammalian nervous system. And in this regard, we've been focusing on the cerebrospinal fluid, or the CSF, and specifically how secreted factors in the CSF, either individually or together, might influence the development of the mammalian brain. And so the CSF actually has a very interesting history. Its function was first described by the early Greeks, who thought it was essentially a connection between the body and mind, and the brain was there to hold it all together. But it wasn't until the Middle Ages, from the work of Da Vinci and Vesalius, where we obtained the first accurate models of the cerebroventricular system. And only about 100 years ago did Harvey Cushing, a pioneer in neurosurgery, demonstrate that the choroid plexus is produced by the choroid plexus, which is a vascularized tissue located in the ventricles in the brain, that this tissue produces the CSF for the brain's ventricles. And so we know today that the CSF fills the brain's ventricles. It flows from the lateral ventricle to the third and the fourth ventricle. It then can go down into the central canal of the spinal cord and into the subarachnoid space, where, together with recently described lymphatics, it's resorbed into the venous circulation. The CSF is very well established in its roles as a fluid cushion for the nervous system. It also maintains ionic balance for the brain, and it's a passive sink for waste and debris. And so it's estimated that an adult human has about 150 mils of CSF in the system at any given time, and the CSF turns over about 3 to 4 times every day. And so this means we're producing upwards of 500 mils or a half a liter of CSF on a daily basis. And so this exchange of CSF has brought about several new hypotheses for the roles and functions for the CSF over the last several years. And some very exciting new roles for the CSF now include roles in the regulation of circadian rhythms and sleep, in neuroblast migration in the adult nervous system. CSF and abnormal CSF levels are implicated in a number of developmental disorders, including autism spectrum disorders and also with mental illnesses. And recently, work from my lab as well as several other labs in the field have explored the idea that the cerebrospinal fluid might provide a stem cell niche for the developing and adult brain. And a stem cell niche would then suggest that the CSF might provide survival, proliferation, and maintenance cues for the mammalian brain. And the work in my lab has focused specifically on the embryonic or the developing nervous system. And one of the key factors that we've worked on is the insulin-like growth factor 2, or IGF-2, shown in the schematic, which we found is secreted by the choroid plexus into the CSF, and then it binds to receptors on the surface of cortical progenitor cells to regulate their division and ultimately brain size. And so we originally became interested in this topic because we have a long-standing interest in wanting to understand the basis for neurologic disease. And it's actually quite remarkable that studies in humans have shown that genetic deletion of single genes involved in cell proliferation during early development can lead to profoundly diminished brain size, as shown in this MRI of an individual, of the individual on the right hand in this image on the slide, compared to a healthy brain on the left. And because many genes that are involved in these processes are highly conserved, it's been possible to model these microcephalies in the research lab. And so these types of studies, in addition to work from a huge number of labs in the field of brain development, have led to a current model of how cell proliferation guides cortical development, which I have summarized in the following slide. And so in the developing brain, for the developing cerebral cortex in particular, the cortical progenitor cells during the earliest stages of brain development are all located in the ventricular zone, which lies immediately adjacent to the brain's lateral ventricles. And the progenitor cells in the ventricular zone undergo a series of lineage restriction steps. And very early on, we have neuroepithelial progenitor cells, which contact the cerebrospinal fluid, and they also extend primary cilia, or these antenna-like extensions, into the fluid. And these neuroepithelial cells later develop into radioglial progenitor cells, which then become further lineage restricted and into other types of progenitor cells for the developing cortex in the brain. But for the purposes of the experiments and the studies that we've been focusing on, we've really looked at the radioglial progenitor cells. And here I have an example of an EM of the ventricular surface of a developing rodent brain, where you can see the primary cilia that these progenitor cells extend into the CSF. And so the location of these progenitor cells and the primary cilia that they extend into the CSF have raised the possibility that perhaps signals and proteins that are distributed in the CSF might also bring instructive cues to these progenitor cells that then, together with the genetic information, regulate the proper development of the brain. And so we developed tools to collect pure samples of CSF from the developing nervous system, and we pair this with a neural progenitor cell culturing system. And in some of our very earliest studies, we actually found that embryonic cerebrospinal fluid is able to really effectively support early brain growth. And so the image on the right shows an explant that's been cultured for 24 hours in pure cerebrospinal fluid. And we can see that this tissue is very healthy. It has a number of healthy developing neurons that are marked by 2 to 1 staining in green, and it has a number of healthy proliferating progenitor cells shown in red. And this is very similar to a freshly prepared specimen of a normal developing brain. And in contrast, if we were to culture these neural progenitor cell cultures in an artificial CSF that contains normal ions and mimics the ionic composition of the CSF but lacks all proteins that are normally present in the CSF, we see that the tissue really doesn't thrive and that the progenitor cells that are normally located along the ventricular surface are not healthy and dividing. And so these studies really demonstrated that the embryonic and the developing CSF harbors important growth and survival promoting signals for the developing brain. And so we started to then explore, well, you know, what is so special about the CSF, and what are some of the proteins that are in the CSF that give it this tremendous ability to promote brain health and development. And so we started to look at just the total protein composition of the CSF, and we found that it varies dynamically with the age of the developing brain, and that its total protein composition peaks around birth in rodents. And similar findings had been previously reported by other groups in other types of species. And this was a rat from a developing rat brain. And if we analyze the CSF by a silver staining method, so across a similar time course, we find that the silver staining pattern shows a dynamic range of proteins that are available in the CSF. And when we look at it carefully, we see that there are proteins that are present early and then disappear. We also see apparent transient appearance of proteins during different stages of development. And we also can find proteins that are present in adults that are not detected during early embryonic development. And so this really shows us that the proteome in the CSF is highly dynamic over the lifespan. But we really wanted to get back to understanding, well, what's so special about the early CSF that can really support and drive the development of the brain that I showed you in the previous slide. And so we started to analyze the CSF from embryonic day 17 developmental stage by mass spectrometry. And in these studies, we identified hundreds of proteins in the CSF, and we were really intrigued by the identification of peptides that corresponded to the growth factor, insulin-like growth factor 2, or IGF-2. And this was really interesting for a number of reasons. IGF signaling had already been implicated in prenatal growth and brain size by several groups, including the work from Arja Stratiotis' lab and Joe Dercole. And the IGF signaling pathway is really well established as a regulator of stem cell proliferation in many biological systems. But the availability and distribution of IGF-2 in the developing brain was poorly understood at the time that we were doing these studies. And so we performed some Western protein analyses, and we found that IGF-2 is transiently available in the developing rodent brain, and it peaks during the time that the cerebral cortex is forming, and then its availability decreases in adulthood. And consistent with an active secretion into the CSF, we find by in situ a hybridization that is highly expressed by choroid plexus epithelial cells, which are the cells that play an important role in secreting different factors into the CSF and for producing the CSF. And so because IGF-2 is then in the CSF, we looked for evidence of receptors on the surface of the cortical progenitor cells to see if they could actually receive the signal from the fluid. And so the active signaling receptor for IGF-2 is the IGF-1 receptor. And on these images, you can see that we could detect a highly enriched IGF-1 receptor signal along the ventricular surface of the developing brain on the left. And on the right, I have higher magnification images to show you that this is not, that this is a specific state, not an edge artifact, and that the IGF-1 receptor signal is really interdigitating with beta-catenin, which is a marker of apical junctions in these cells. We then looked for the presence of IGF-2 on the surface of cells, and we found by EM that IGF-2 coats the surface of the cortical progenitor cells, as shown here by these dark black packets that are binding to the surface of the cell on the right. And we also observed that the IGF-2 binding was highly enriched on primary cilia that extend into the CSF, as I showed you in the previous slide. And so given that IGF-2 coats the surface of cells and the cells have the receptor, is there then a functional interaction? And so we took advantage of antibodies that have been developed to evaluate the activity of signaling pathways in cells. And so in the classic IGF signaling pathway, when IGF-2 binds to its IGF-1 receptor, this induces the dimerization of the receptor. It leads to autophosphorylation events on the cytoplasmic domain of the receptor, and these phosphorylated residues act as docking sites for adapter proteins and other proteins of downstream signaling pathways that then lead to the activation of a variety of kinases. And in the schematic, I've outlined MAP kinase and AKT, which in turn get phosphorylated when they're activated. And so we took some of the developing CSF and we treated cultured progenitor cells with the CSF, and we found that the CSF was able to activate the phosphorylation of the IGF-1 receptor, as shown in the uppermost blot on the slide, as well as its downstream signaling pathways, AKT and MAP kinase. And so this is really exciting because it really shows us that the CSF is able to interact with and deliver signaling activities to the developing progenitor cells. And it really then suggested that maybe by modulating the availability of these different signaling activities in the CSF, we could push and pull the division of these cortical progenitor cells and possibly modulate brain development. And so we did a series of gain of function and loss of function experiments. We took our cortical explant prep, and we cultured this cortical progenitor cell preparation in basal conditions with supplemental IGF-2, and we found that IGF-2 alone was able to stimulate division of these progenitor cells. We then did a converse experiment where we removed endogenous IGF-2 from developing CSF, when we found that reduction of IGF-2 also then, as expected, reduced the ability of the native CSF to stimulate proliferation. And finally, we obtained IGF-2 knockout mice, which had been generated by Arja Stratiotis' lab, and we collected CSF from these mice, and we found that this IGF-2 deficient CSF had a reduced ability to stimulate proliferation of cortical progenitor cells compared to wild-type control CSF. And so together, these data really demonstrate that CSF IGF-2 can regulate the proliferation of cortical progenitor cells. And so we took advantage of several other mouse models that have been available for the study of IGF signaling in this model. And one example is the IGF-1 receptor. And when we conditionally deleted the IGF-1 receptor from these same cortical progenitor cells, we were able to observe a small brain phenotype that came about from a reduction in the numbers of progenitor cells undergoing cell division. And we also had the IGF-2 knockout mouse. And when we did an analysis of IGF-2 brain development, we found that it had a small brain compared to its wild-type control. And when we analyzed the development of the neurons in the more mature brain, we found that there were reduced numbers of neurons that populated the uppermost layers of the cerebral cortex. Again, showing that IGF-2 and signaling through the IGF-1 receptor at the cortical progenitor cell that lies adjacent to the CSF is important for regulating brain development. And so the data that I've shown you thus far really suggests that then the CSF provides a proliferative niche for neural progenitor cells. And we focused on IGF-2 as one important factor that's secreted by the choroid plexus into the CSF. It binds to its receptors on cortical progenitor cells, and it regulates their division and ultimately brain size. One really fun side project that we had developed into a larger project as well, but it was to try to investigate how CSF and delivery of signals by the CSF might be co-opted by neurologic disease, or in conditions where different secreted factors might be abundantly available in the CSF. And one candidate disease that we thought we would test is a brain tumor condition called glioblastoma multiforme. And these are very devastating brain tumors. They're highly infiltrative, and we have very few therapeutic opportunities for treating these conditions. And so we collaborated with a neuro-oncologist at the Beth Israel Deaconess Medical Center, Eric Wang, who specializes in treating patients with glioblastoma. And he has CSF samples from these individuals he shared with us. We were able to obtain 56 CSF samples from a total of 21 patients and 8 disease-free controls, and we evaluated the levels of IGF-2 in the CSF of these patients. And we found that patients with the glioblastoma brain tumors had significantly higher levels of CSF IGF-2 compared to disease-free controls. And actually, if we looked at the CSF samples from the patients with the highest IGF-2, we also found that these individuals also had the most advanced and aggressive disease. And the individuals with the lowest CSF IGF-2 levels had more benign or early or stable glioma. So these data really then suggest that CSF IGF-2 can also associate with disease progression. And in experiments that I don't have time to share with you today, we also tested the ability of this human CSF to stimulate proliferation and promote health of our cortical explant in vitro cultures, and we found that they were able to promote proliferation in an IGF-2 dependent manner. And so we're really interested in exploring how CSF signaling plays an active role in maintaining health of the brain during development as well as in disease. And this is really important from the very earliest stages of neural tube closure, where neural tube defects can have devastating consequences on normal nervous system development. An example is spina bifida, where we continue to be interested in how CSF is regulated during cerebral cortical development, and I've just shown you some of our data and our projects involving cerebral cortical development and microcephaly. And ultimately, we're very interested in expanding these research directions to understanding diseases in the mature nervous system, in particular how it relates to hydrocephalus, which is a condition in which there's excess CSF in the nervous system, and in other types of tumors, for example, choroid plexus tumors. And so for the second part of my talk, I thought that I would just touch on some recent data in the lab in which we've tested how the availability of important CSF factors might be regulated. Because in the first part of my talk, I showed you how IGF-2 is secreted by the choroid plexus into the CSF, and it regulates important cell developmental processes in the developing brain. But actually, we know surprisingly little about the mechanisms, about the molecular and genetic mechanisms that regulate then the availability of these factors in the CSF. And so we turned to the choroid plexus, which is this highly vascularized sheet of epithelial cells that's located in each ventricle in the brain. It consists of choroid plexus epithelial cells that surround a choroid plexus mesenchyme and a choroid plexus vasculature. And when we were starting these studies, it was clear that the choroid plexus is very important for producing CSF, but it's typically grouped so that each each ventricle's choroid plexus is sort of considered to be very similar to each other. And also, typically, the focus of other studies really has often been on the lateral ventricle or the fourth ventricle, and not really a comparison of different functions that the choroid plexus tissues in the different ventricles may have. And so we started off by just testing if the CSF in the brain's ventricles could be different, or is it all the same. And on the right, we have a silver stain that shows that while there are many proteins that are in common between the CSF and these different ventricles, there are actually also very apparent differences, that there are bands that show up in the lateral ventricle CSF, and then there are other bands that are more prominent in the fourth ventricle CSF. So these initial data then suggested to us that maybe there's a regionalization of the CSF in the nervous system that really hasn't been previously explored very, very well. And so we decided to analyze the choroid plexus in the different ventricles in the brain. And we isolated the rodent choroid plexus from the lateral ventricle and fourth ventricle, and we decided to also FACS purify just the epithelial cells in the choroid plexus. And this means we're purifying and isolating just the cell population that's considered to secrete the CSF for the ventricles. And we applied a technique called RNA sequencing, which allows us to analyze all the transcripts of all the transcribed genes in the tissue. And we identified thousands of genes that are expressed in the tissues, and this heat map actually displays the significantly differentially expressed genes between the different tissues. So we found that there were hundreds of genes that were differentially expressed between the lateral ventricle and the fourth ventricle, and we also identified genes that were differentially expressed by the epithelial cells versus the stroma and the vasculature. And here's just an immunostaining example of some of the genes that were highly differentially expressed by the different cell types in the choroid plexus. On the top, we have an example of PCAM, which marks endothelial cells, and on the bottom, we have an example of FOXJ1, which is a transcription factor that's highly expressed in the epithelial cells. So we did some additional analysis of the different genes and the differential expression patterns of these genes. And our analyses revealed that the different choroid plexus tissues were really not uniform, but they really retain these transcriptional memories of positional identities that really reflected their origins in very early stages of organ and body development. And they really went back and expressed genes that are normally thought to be expressed at specific sites in the axial nervous system. And so, for example, in the case of the fourth ventricle, previous work from Sue DeMecky's lab here at Harvard Medical School has shown that different homeobox or HOX genes are expressed in the fourth ventricle choroid plexus. And we similarly observed that many of these HOX genes were expressed in the fourth ventricle choroid plexus, but they were essentially absent in the lateral ventricle choroid plexus. And we found similar and opposite effects for a number of other genes, and here we have a few examples where EMX263 and OTX1 were more abundantly expressed in the lateral ventricle versus GRAIL2, MEIS1, and HOXA2, for example, were genes that marked identity of the fourth ventricle choroid plexus. And these differences in gene expression actually translated to differences in protein expression. And so on this slide, I have a few representative images of choroid plexus epithelial cells showing that these different proteins are differentially present in the lateral and the fourth ventricle choroid plexus as well. And finally, we were really interested to test if this might lead to a regionalized CSF proteome. And so we collaborated with our colleague Hanno Steen here in the Department of Pathology at Children's Hospital to perform some quantitative mass spec on choroid plexus conditioned media. And so what we did was we isolated the lateral or the fourth ventricle choroid plexus, and we cultured these tissues for 24 hours, and we analyzed them by mass spectrometry. And we found hundreds of proteins that were differentially secreted by these different tissues into the culture medium. And this was very exciting for us because it really proved that this different transcriptional signature that the tissues have that originates at the very earliest stages of body development then translates to the production of regionalized CSF proteomes in the more mature brain. And finally, we started to look to see if these differences in protein availability hold true throughout life. And so, as I mentioned before, these initial studies were performed in the developing brain. And here we have example of quantitative PCR from the choroid plexus, from either the lateral or fourth ventricle. And we find that actually many of these patterns of gene expression change between the developing and adult brain. And this also translates to the availability of proteins in the CSF, as shown by these Western blots on the right. And so this brings us to our summary slide, where I hope in the first half of my talk I convinced you that the embryonic CSF provides a proliferative niche for neural progenitor cells, and I focused on IGF-2 as one example factor that's released and secreted and released by the choroid plexus into the CSF. It binds to its receptors on the surface of cortical progenitor cells and then regulates their division and brain development. I also then touched on the role that IGF-2 may have on disease progression in glioblastoma multiforme, which is a devastating brain cancer. And in the second part of my talk, I showed you how some of the approaches we've taken to begin to analyze how these important proteins in the CSF are regulated and produced by the choroid plexus tissues that are located in the brain's ventricles. We used RNA sequencing to define, and together with quantitative proteomics, to define a choroid plexus secretome. And we've identified a regional identity in the choroid plexus tissue that parallels its tissue of origin. And we've also described a region-specific CSF secretion by these different choroid plexus tissues. And so that brings me to my acknowledgement slide. And so I'd really like to thank all the members of my lab, past and present, who've worked tremendously hard on the various projects that I've presented here and also on ongoing work in the lab. The second part of my talk was performed largely by Melody Lunn, who is an MD PhD student in the lab. We're also indebted to wonderful collaborators who dedicate their time and resources, and I mentioned many of them during my talk, Eric Wang, and also many of these experiments were initiated when I was a postdoctoral fellow in Chris Walsh's lab. And there I had the opportunity to work with a really talented group of postdocs and graduate students, Mauro, Jenny, Shi, and Matt, to name a few. We've done our quantitative proteomics in collaboration with Hanno Steen and Kevin Broadbelt. And we've done a lot of collaborations with Hart and Monica to analyze how these patterns of gene expression are conserved in human, across rodent and human systems. We collaborate also with Anthony Lamontia and Tom Maynard, who have been really helpful with establishing these cortical explant systems and helping us develop tools and analysis methods for the CSF. The RNA transcriptome and FACS experiments were performed in collaboration with Ed Manucci at UC Irvine, and we've also collaborated with Nenad Sestan to obtain samples to analyze the evolutionary, the nature of the gene expression patterns and to test how they're evolutionarily conserved. And we've collaborated also with Michael Holtzman and Yang Zhang at Washington University to analyze the region-specific availability and expression of proteins in the CSF. And so we're tremendously grateful for funding that has supported our work, the Alfred P. Sloan Foundation, the Pediatric Hydrocephalus Foundation, the NIH, as well as Boston Children's Hospital. Thank you. And I'll pass it back to Susan at this time. Thanks.
Thank you very much, Dr. Laitman. As a reminder, please continue to submit your questions for the speakers via the question box, and we will address these during the Q&A session following the second presentation. Now I'd like to welcome our next speaker, Dr. Wang. Dr. Wang did his graduate study at Harvard University, where he studied the development of the Drosophila visual system. During this period, he published several high-profile papers, including 2 first-authored papers in Cell. After finishing his graduate work, Dr. Wang switched to the study of neural development in the mammalian system. He worked with Dr. Louis Reichart at UCSF during this period and was supported by a fellowship from the Helen Hay Whitney Foundation. After starting his own lab at the University of Wisconsin-Madison, Dr. Wang became interested in neurovascular interaction in the developing brain. Among others, he has discovered a novel role of neural progenitor cells in regulating blood vessel stabilization in the developing brain. His research is being supported by a Basil O'Connell Award from the March of Dimes Foundation and grants from the NIH. Today, he will discuss his findings in this area. Dr. Wang?
Thank you, Susan, for the nice introduction, and hi, everyone. So today I'm going to talk about neurovascular interaction in the brain, especially in the developing brain. So when we think about the brain, the first thing that comes to mind is usually the neurons, the firing of the neurons, and transmitting of neural information. But there's actually a vascular and intricate network of blood vessels in the brain that is critical to supply nutrients and oxygen to the brain. And as we know, probably one of the most common brain disease is actually stroke, which is the malfunction mostly of the vascular system of the brain. So the neurons in the brain, as you may remember a little bit from Maria's presentation, they are born by these neuroprogenitor cells called radioglia of the brain, and these cells, they derive from the neuroepithelial cells at the early stage. And then these radioglia cells then gave birth in a serotypic order, first of neurons, and then of later developmental stage of exercise and oligodendrocytes. And these radioglia cells also provide as a scaffold, a structural scaffold for the migration of the neurons. So radioglia really plays an important, essential role in the development of the neural components of the brain. And the vascular system, some of you may not be as familiar, but it is also a very elaborate network as shown in this slide. On the left side is on a surface view of the pure surface of the brain, and you can see there's elaborate network of large vessels. Then on the right panel, you can see these vessels branch and dive into the brain parenchyma itself, and then they branch into even smaller microvasculatures. And these are the blood vessels that really supply the nutrients and oxygens for the brain to properly function during much of the adult life. So how this network developed in a rodent brain, you can see on these slides, there are from the early stage, actually all these vessels of the nervous system, they are produced in a process called sprouting angiogenesis. They all originate from outside the nervous system. And then, so in panel A, you can see that initially with very few vessels, and then they begin to elaborate as development goes on. From panel B to D, you can see the gradual elaboration, and from the quantification, you can see in panel E, is initially at E13.5, it is very low vessel density, and it gradually increases and plateau around E15.5. This is in a rodent brain. And also, you can see the branching also stabilize during the developmental processes. So as you may have noticed in the previous slide, there's actually been noticed a long time ago that the neuroprogenitor cells or the radioglia, they actually interact with the blood vessels throughout development from the very early stage and later stages. And we have also been able to confirm this by 3D confocal imaging, and as shown in this figure, you can see that there is a direct interaction of radioglia cells as labeled in red here, and with the endothelial cells labeled by IB4 staining, and you can see this direct contact area is actually an area where there is no parasite coverage. So the parasites are labeled by NG2 in blue, and these are the support cell, vascular support cells in the brain. So you see there's a direct interaction. So what I'm going to talk to you briefly today is about an experiment we've done that show that actually there is a radioglia has a novel role in regulating the stabilization of blood vessels in a nascent developing brain. And so on the left side, I show you previous results have shown that there's a canonical Wang signaling plays a very important role in initial vessel ingression into the brain throughout both the forebrain and also the spinal cord as well. And we have found, and this is what I'm going to hope we will convince you, that there is actually a second step. That is, in that step, the radioglial cells actually downregulate these canonical Wang signaling in endothelial cells, and that help stabilize the nascent blood vessels in the brain. So the experiment actually was started when we were studying a function of a gene called OCT3, which is a subunit of the complex called origin recognition complex, which is a complex essential for DNA replication and cell division. So you don't need to remember all the detail. It's just we use this as a genetic tool to ablate cells, because particularly dividing cells, because this complex is essential for cell division. So if we use conditional allele of the OCT3 and induce it using a Cre line into dividing cells, we will block the cell division of that cell population, and in a way we basically ablate that cell population. So the Cre lines we use is a nesting Cre line generated by the Klein lab, and it has been shown before is specific to neural cells, as can show in these two rows of images of two different stages, E16.5 and E17.5. And this is crossed to a reporter line, then which express EGLP when the reporter is recombined, but when it's not recombined, it will express M tomato. So you can see in G, for example, you can see the green neural cells that are recombined by this Cre line, but in G prime, you see the vasculature that is not recombined. So we use this line to induce the OCT3 gene deletion and block cell division on the neuroprogenitor cells. So as you can see in this slide, at the E13.5 in panels A and B, we didn't see much difference in proliferation yet by the BRDU staining, but by E15.5 in panels C and D, you see the beginning of the reduction in cell division. And if you look at the radioglial cell density as labeled in E and F at the stage of E15.5, see the beginning of the depletion of radioglial cells, and then by P0 in panels G and H, you see a very dramatic loss of the radioglial cells at this stage. So as I mentioned earlier on, so the radioglial is really essential for the production of neurons and neuronal migration, and so we were not surprised to find that the brains are smaller in these animals. But what was most surprising to us was that actually we saw a dramatic defect in blood vessel formation in these mutant brains. So as shown here in panel A, you see the hemorrhage of the brain, there's bleeding. And then in CD and EF, we use two different markers to label the vasculature of the neonatal brain, and you can see in D and F in mutants, there's virtually no blood vessels left, and these are quantified in G and H. You can see, again, the dramatic loss of blood vessels. So there was some concern about whether these defects could be due to as I mentioned, the neuronal production or migration defects. So we actually use several mouse mutant lines, including beta-1 integrin in panel B and BRCA1 in panel C. These two mutants each have either a neuronal migration defect all the neuronal production defects. So as we can see in panels B and C, neither mutation alone was sufficient to produce any vascular defects. And in D, we actually also generated a double mutant of these. So we have basically mimicking the neuronal defects in OC3 nesting Cree mutants, but we did not see any obvious vascular phenotype, as these are quantified in panels E and F. There's basically no defects in either vessel density or branching frequency. So this convince us that the defect is actually because of the depletion of the radial glial cells. Again, this conclusion was also supported by the finding that we have another mutant generated using a different Cree, the HGFAP Cree, which comes on slightly later. So as is shown in this slide, we have the nesting Cree mutant in yellow, and then the HGFAP Cree in purple. You can see there's a close correlation between the loss of radial glial and the severity of the phenotype in both of the reduction in vessel density and reduction in branch point frequency. So the more radial glial you lose, the more severe is the phenotype. So this again argues that the radial glial may play a direct role in regulating blood vessel development in embryonic brain. So how does this phenotype develop in the mutants? So we actually look at the phenotype throughout the developmental stages. So as shown here, we look at from E15.5 in A and B to E18.5 G, as you can tell, the phenotype actually first appear around E16.5 in panels C and D, and quantified in I and J. You can see the initial reduction at E16.5, but before that, at E15.5, actually all the vessel morphology and density and branching all looks fine. We also even normalize the volume of the vessel density against the volume, and we still see that reduction at E16.5, suggesting that this is really a regression phenotype. Basically, that means is the vessels initially form normally, and then the network begins to break down after the loss of radial glia. So another piece of evidence for that argument is also we see the so-called collagen 4 sleeve. So these are collagen 4 are basically a component of the basement membrane, and normally they line the blood vessels uniformly as shown in P prime. The red ink is collagen 4, and in Q prime you can see the collagen 4 pointed by the arrowhead. You see the collagen 4 staining, but there's no vascular cells and associated with the collagen. So this is the so-called collagen sleeves, which are indications of vessel regression. So we were convinced that this is a vessel regression phenotype. Now the question is, how does radial glia regulate the vessel stabilization, and why their absence results in the regression of blood vessels? So one possibility is certainly the support cells, the pericytes I mentioned earlier. They are known to be critical for vessel stability. We quantify the pericyte density using a number of markers as desmin in KNL, and then PDGFR beta in M and N. As shown in panel O, you can see the pericyte density is not affected by the mutant. So although there's possibility that there might be finer structural or interaction defects, we think that these pericytes does not seem to play a major role in this vessel regression phenotype. So we basically move on to look at other pathways. One of the pathways we look at actually is a canonical Wing signaling. As I mentioned, is critical for initial vessel regression into the neural tube at the early stages of development. So here is shown one of the published work that's showing the double deletion of wing 7A and B from neural cells, as in panels E to H and I to L. You can see the resulting hemorrhage throughout the central nervous system, and also the lack of vessels in the nervous system in H and L. You can see there's very little staining in comparison to the B, that's the wild-type control. So very interestingly, if you look at the ligand expression, the Wnt7A and B expression throughout development, you can see early on at E15.5, both are more or less, particularly E7A, they are expressed in radial glial cells, and N7B also in neurons. But at later stage, at E18.5, they both are expressed in cortical neurons. So this indicates the ligand is actually always there. But if you look at the pathway activity, so using a beta-catenin-driven LAG3 reporter, you can see as we quantify in panel E, at early stages, there's a very high density of LAG3 positive cells. But then as development goes on, there's actually a reduction, gradual reduction, and a downregulation of these Wnt signaling pathway. This suggests to us that maybe this suggests that the Wnt pathway is dynamically regulated during development, and that this downregulation may also be important for vessel development. So we actually look at the Wnt pathway activity in our mutants, and as shown here in G and H. So we look at the blue cells along the blood vessels, as shown here, indicated here by the by the white arrowheads. We actually see a dramatic increase in the number of blue cells that are along the vessels, and as quantified in L, there's almost a tripling of the LAG3 positive endothelial cells. So this suggests to us that actually the Wnt signaling fails to be downregulated. So normally they are downregulated, but in this case they remain high in the mutant. So we also have look at another Wnt reporter, a group 1 gene that is known to be regulated by canonical Wnt signaling, as shown here. We look at both immunostaining in E' and F'. There's increases in staining as well as in Western blot in I and J. You can see the group 1 also is upregulated in mutants. So this again argues that unlike in a wild-type brain where Wnt signaling is downregulated in endothelial cells, in our mutants, the signaling remains high. So the question becomes is whether this plays a role in the phenotype we observe, in the vessel regression phenotype we observe. So we have also some in vitro culture data shown in this slide. I'm not going into the details for your best, just to show you in panel C that we quantify the Wnt signaling in endothelial cells, in cells that are contacting radioglial or those that do not contacting radioglial signaling in culture, and we see that when endothelial cells are in contact with radioglial, they typically have very low level of Wnt signaling. So this again argues that there is a suppressing signal from radioglial that may normally downregulate Wnt signaling in endothelial cells. So now, as I mentioned earlier, our question now becomes is whether this high level of Wnt signaling is responsible, at least in part, for the vessel regression phenotype we observed. So to test this, I'm going to tell you about two sets of experiment we did. One is to see whether we can mimic the effect, and the second is whether we can suppress the effect in our mutants. So just to summarize quickly, what I've been saying is our hypothesis is that in a normal brain, in the early stages of vessel development, the Wnt signaling is critical for the initial vessel ingression into the brain, but at the later stages, radioglial actually provides an inhibitory signal in here as represented by a red bar, and that is responsible for downregulating Wnt signaling in the endothelial cells. But in our mutants, we have the ablation of radioglial, and that results in a release of these suppression. So the Wing target genes, including LAG-Z, group 1, and presumably some other critical genes that are upregulated and that we believe may result in vessel regression. So the question is now, can we mimic this condition in a wild-type brain? So the approach we've taken is we're going to treat the brains with some GSK inhibitors such as lithium chloride and ask whether we can upregulate Wing signaling in a normal brain and then also mimic the vessel regression phenotype we observe in our mutants. So first we look at the reporters for the Wing pathway activity as shown here in panel A and B is for the LAG-Z staining, and then D and E is the group 1 staining. You can see lithium chloride indeed can upregulate the Wing signaling in the wild-type brain and also in the group 1 expression in the endothelial cells. So this indicates that our condition is working. Now we look at the brain, and we found that indeed this treatment results in a mild but widespread hemorrhage in the wild-type brain as shown in panel A. You can see the lithium chloride treated brain has this reddish color, and in C and D you can see there's actually a mild vessel regression phenotype. And we quantify the result and show that in panel B, this is phenotype actually very specific to the cerebral cortex. We do not observe these changes in vessel density in the striatum or in other regions, other organs of the body. So this suggests to us that the abnormally high level of Wing signaling in the brain actually may result in vessel regression. So too much Wing signaling actually is not a good thing for the blood vessels in the brain. And so the second approach we've taken is to see whether, so we are saying that there's too much Wing signaling in the endothelial cells. So the question is, if there's a way to downregulate Wing signaling, can we suppress the phenotype we observed in our mutants? So again, here is a summary of our model that with the ablation of radial glia, we have the release of the suppression on the Wing signaling and resulting in some genes that presumably lead to the vessel regression. So what we want to do is, can we somehow say, take away some of the Wing ligands and then downregulate the Wing target genes? Could we suppress the vessel regression phenotype we have observed? So as I mentioned, because Wing 7A and B, they are required for initial vessel regression, we cannot really do the experiment by taking both away. So the way we try to do it is to take one of the ligand away, Wing 7B away, and it's been shown previously that Wing 7A or B single mutants actually don't have a vessel ingression phenotype, so that's good for us. So the first thing we did, then we try to gauge whether the removal of Wing 7B can indeed attenuate Wing signaling in our mutants. So here we use a group 1 as a reporter for the Wing pathway activity. So as is shown in panel E, in the control, we have a moderate level of group 1 expression. In our mutant, OX3 mutants, we see a heightened level of group 1 expression, indicating higher level of group Wing signaling in a double mutant. So basically, we remove Wing 7B from neural cells in the OX3 mutant background, and we end up having a lower level of group 1 expression. So this is quantified in panel F. You can see there's a significant suppression of Wing signaling in the endothelial cells and the brain. So now the question is, can we suppress the phenotype? So indeed, we found a milder hemorrhage as shown in panel E. In the double mutants, we generally have a smaller area of hemorrhage in the mutant brain, and in panels G and H, we saw a modest but significant restoration of the vessel density in the mutant brain. So this is quantified again in F. As the vessel density you see in the control or the Wing 7B single mutants has no effect on the blood vessel development. In the OX3 mutant, single mutants, we saw a dramatic reduction in the vessel density, but when we remove Wnt7b from that mutant background and thereby attenuating Wnt signaling in the endothelial cells, we saw partially some of the vessels in the mutant brain. So this argues that Wnt7b or Wnt signaling upregulation is at least partially responsible for the vessel regression phenotype we observed in the after radioglial ablation. So just to summarize what I have told you, so we found that there is a direct interaction of the radioglial, the neuroprogenitors of the brain, with blood vessels from the very beginning of brain angiogenesis, and then radioglial actually play a very critical role in the stabilization of the nascent vessels, and we believe this is through the inhibition or downregulation of canonical Wnt signaling in the endothelial cells. And we have also data showing, which I did not talk about, is that this part of this it could be through downregulation of some of the Wnt target genes such as MMPs, which may be critical for blood vessel stabilization. Again, so just to summarize, just to give you a picture representation of what I said, in the early stages, several labs have shown that as a critical role of Wnt7A and B, canonical Wnt signaling in two endothelial cells that are critical for vessel ingression into the brain, and at later stages of brain angiogenesis, we believe that there is a second step where the radioglia actually plays a role in downregulating canonical Wnt signaling in endothelial cells, and this is critical for the stabilization of the nascent vessels. So now the one crucial question, of course, is what is a signal? So I'm just going to briefly mention what we've been working on in the past year or so. So there's this is work still in progress. So one area we've been working on is understanding the epigenetic changes in endothelial cells in the brain. So we have evidence now showing that these changes in the Wnt signaling capacity in the endothelial cells is actually epigenetic. So if you culture cells from different stages of the you culture endothelial cells from different stages of brain development, and you use, for example, artificial methods to activate Wnt signaling, you can actually find that these cells at different stages, they respond very differently. And we can passage these cells in vitro for several generations, and these differences still stay the same. So this suggests there's actually some epigenetic changes that take place during development in the endothelial cells. And so we are now trying to figure out what that changes are and what signaling pathway could trigger that change. So that's one direction we've been working on. A second direction we've been working on is to try to profile gene expression changes in endothelial cells in our mutants. So for that purpose, we have actually generated a so-called trap transgenic line. Basically, it's translating ribosome affinity purification method, where we label the ribosomes in endothelial cells, and then we can purify the mRNAs from the endothelial cells from the control and mutant and profile the changes in the gene expression. We actually have gone through the RNA-seq and found some very interesting changes in gene expression and working on some candidates. So I hope we'll, in a not too far future, I can tell you more about this. So last, I just want to acknowledge people and our collaborators on this work. So this work has been done by several graduate students and postdocs in my lab, including Sharon Ma, who's done the most work on this. And then currently, David Sansosh and Piyush Kumar has been continuing working on this project, and work was done in collaboration with Lou Reichert at UCSF, and this work also supported by the Marshall Dimes Foundation and the NIH, AHA, and UW Medicine. And thank you for your attention. Now I pass it back on to Susan.
Thank you very much, Dr. Wang. We'll now begin the Q&A session. Please continue to submit your questions for the speakers. Dr. Laitman, how is Ig2 normally regulated? Or IGF-2?
Yeah, so that's a really good and important question. So IGF-2 is a gene that's known to play really important roles during the development of the embryo and during early fetal development. And it's typically imprinted such that the paternal allele of IGF-2 is expressed in almost all tissues. But there are several areas in the body, including choroid plexus, where it's biallelically expressed. And so that's been a really interesting piece of information. And right now we're still trying to understand exactly how IGF-2 is regulated at the choroid plexus. Our RNA sequencing data has shed light on which forms of IGF-2 and which promoters are active to regulate IGF-2 expression in the choroid plexus during brain development, but we still have a lot of work to do to uncover the exact mechanisms.
Thank you. Dr. Wang, how similar is human brain angiogenesis to rodent angiogenesis, and are there other key signaling pathways that are involved other than what you described with Wnt?
So the early steps of brain angiogenesis is believed to be very similar between the mouse brain and the human brain. But of course, the human brain is a lot bigger, so presumably there's a more complicated regulation ongoing. So besides the canonical Wnt signaling, so this area is actually just beginning to be studied. So there actually in rodents, there's evidence actually TGF-beta signaling also plays a very important role, particularly so here is actually a slightly different regulation where the radioglial cells also there's a cell adhesion molecule called beta 8 integrin that actually locally regulate the TGF-beta activation, and that is being shown to be critical for the integrity of blood vessels.
Thank you. Dr. Laitman, you described some differences between the fourth ventricle and the lateral ventricles. Why is that important in cortical development?
This is also a really interesting avenue of research that we're continuing to pursue in the lab. So some of the differences and factors that are differentially secreted by the choroid plexus tissues include growth factors and morphogens, such as sonic hedgehog, which is really important for hindbrain development and cerebellar development, and choroid plexus proliferation in the fourth ventricle is abundantly, very, very highly secreted by the fourth ventricle choroid plexus, and we don't see that expression in the lateral ventricle. But then there are other factors, certain family members from the BMPs, for example, bone morphogenic proteins, which are more abundantly expressed by the lateral ventricle choroid plexus. And we hypothesize that the differential secretion of these factors during early stages of brain development should inform or instruct the development of adjacent tissues, perhaps in the patterning or other aspects of their development. But it's very important to remember that the CSF is flowing as well, and so there's going to be mixing of these factors. And right now, experiments are ongoing in the lab to try to understand the exact functional consequences of this regionalization.
Thank you. Dr. Wang, how does the research that you described, how does that relate to human disease? So one of the questions here is, in diseases such as atriovenous malformation, can the loss of capillaries be associated with some of the mechanisms that you described?
So there are actually several diseases that's more related, closer related to neurovascular interaction. For example, in a disease called germinal matrix hemorrhage, which is basically a very common condition in neonates, where there's a bleeding in the specific region of the brain where So that can cause conditions such as cerebral palsy and hydrocephalus. And so in those, it's been found that the blood vessel integrity in that brain region is really different than other brain regions. And so we actually have a little bit work on that, showing that there actually a brain region specific signaling pathway from radial glia to the blood vessels. And in this arteriovenous malformation, also there is the evidence in implicating the TGF-beta signaling, as I mentioned earlier, that is also being found to be critical for vessel integrity, and there's some evidence implicating that signaling pathway may be involved.
Thank you. Dr. Laitman, there's been some recent research that's shown a link between the gut microbiome and the brain. Do you think that one way that this could be connected is through the CSF? Do you think that gut microbes could be influencing signaling molecules found in the CSF?
I think that's a really interesting and intriguing possibility. Because there is a connection, because the choroid plexus is highly innervated by it's highly vascularized, and so there's a lot of blood and systemic signals make their way to the choroid plexus. And while we've been thinking about the epithelial cells as secreting proteins into the CSF, these epithelial cells also have receptors, and they're receiving signals from the blood side. And so anything from the body that might find its way into the bloodstream I could envision how it could have impact on choroid plexus gene expression at the epithelial cells. So I wouldn't rule it out. It would be a really interesting possibility to test.
Thank you. Dr. Wang, are there any cell-cell communication signaling molecules such as adhesion or ECM that play a role in neurovascular development?
Of neurovascular. So, as I mentioned, so one of the probably best studied cases in this interaction, in particular in the developing brain, is the TGF-beta signaling, actually. So the TGF-beta, the ligand itself, actually is normally expressed in a latent form. So it's not active, but so the activation process actually dependent on the function of the neuroprogenitor cells, or the radial glia. So in this case, the radial glia provide a key component called a alpha V beta A integrin, and that is critical for the cleavage of the latent TGF-beta. And so with the cleavage, then the TGF-beta can be locally activated. So this is in a way of regulating the signaling to the blood vessels, even though the ligand itself is not provided by the neuroprogenitor cells, but the activity regulation is done by the neuroprogenitor cells. So in a way, it's the same as a regulation of neural to vascular signaling, basically.
Thank you. Dr. Laitman, have you discovered any epigenetic factors that are involved in this CSF signaling?
That's a really interesting question, and it's something that we've thought about, but we haven't. Our work hasn't really touched on that up to now.
Okay, thank you. Dr. Wang, could you go into some detail on where the blood vessels in the brain, where they come from? Or somebody asked if they're of mesodermal origin.
Well, they originally they are mesodermal origin, I believe. So they are the same origin as all the blood vessels in the body, but then they basically have to go through the basement membrane that surrounds the neural tube. So the neural tube initially is just one layer of neuroepithelial cells. The apical side facing inside the tube, and then the basal side is supported by a layer of basement membrane. So as all epithelial cells, and then so inside that neural tube, there's basically initially there's no endothelial cells. So all the endothelial cells have come from outside by sprouting angiogenesis, and then they elaborate in the brain.
Okay, thank you. Dr. Laitman, could you go into a little bit of detail about some of the evidence for the relationship between changes in CSF and autism? Somebody asked if there was a relationship between the basal metabolism rate and autism.
So this is some of this is very recent work where it's been shown that increased levels of extraaxial CSF. So this is CSF that surrounds the brain during different developmental time periods can correlate with the onset of and diagnosis of autism later in development. And there's a paper, Shen et al., I believe in Brain Research, which is a very nice study that has looked at this. And I'm sorry, what was the second part of your question?
They had asked if there was any potential relationship between CSF and the basal metabolism rate in autism.
It may be possible. I'm unfortunately not sure about that, but we'd have to look it up on PubMed. Thanks.
Okay, thank you. And Dr. Wang, will ask you the last question. Does Wnt signaling have anything to do with chemokine receptors and their interaction with endothelial cells?
Actually, I'm not sure about the answer to the question. So, yeah, sorry, I am not familiar with this chemokine signaling.
Okay. Okay, thank you very much. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are displayed on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today and for sharing your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. Laitman and Dr. Wang, as well as our webinar sponsors, BioLegend and Neuroscientist News. Thank you, everyone, and goodbye.